So we are to discuss applications of biosafety. Now, applications of biosafety basically designs procedures, rules, guidelines, and regulations with respect to how you can make the risk level zero, almost nil, or negligible, or if the risk level is very high, how are you going to protect the person working in the laboratory. So that is why it is dealing with a lot of procedures, protocols, which are safety protocols. And that is where biosafety works. So these safety protocols, so please bear with me. The network is really not smooth today.
हम लोग को प्राथमिक चरण से दूर करे जनता को नाम है प्रत्येक को सुधार के यूज करे तो ऐसा कुछ प्रमाण
Again, once again, sorry. So coming back to the no topic. So safety protocols, they are designed for other areas other than our biology also. Very easy to understand is the fire safety manual, or easy to note is the safety manuals used for chemical industries. They also deal with it, but here we are concerned with those people who are working with pathogenic agents in the laboratory. So that is why biosafety has to deal with, if you can read it in the second line or second point over here, it has to do something with disease causing organisms. It has to do something with those organisms with which we are doing our rDNA technology. We are going to go for genetic modifications into the laboratory. So it's more concerned about procedures and rules with reference to this kind of organisms. So what with what all else it's dealing with. So I go with the points as described in the slide. So it is the one which will design rules, regulations, because you want to protect animals, plants from disease causing organisms in different kind of situations. Also you want to protect human life from risk arising due to additives, contaminants, residual toxins, again the organisms which may be present in food, beverages, in your laboratory, and to avoid risk organisms movement. So one term used here is transboundary movement. So transboundary movement means the movement of organism between the states, between the countries, between the towns. That is what is transboundary movement. So if your lab is somewhere based, let's assume it's somewhere based in Delhi and you're working with BSL 4 level organism. So level 4 organism are considered to be the most risky organisms. They do not have any kind of treatment. If they infect you, you have no cure possible. So it's a very deadly lethal organism. Now that lab is somewhere based in Delhi, and you are transferring those organisms, or by chance you have to supply a kind of culture of that organism to some other lab which is in Bangalore. So in between whatsoever cities you are crossing through, because they are not the places where these organisms should be exposed. It should only be exposed in DSL 4 lab infrastructure only, because there you have the containment established, which is going to become the protective mechanism against these organisms. So obviously, in between you can't allow the organisms to escape, or you shouldn't release. So to avoid this risk organisms in transboundary movement, you need to have defined protocols for even transport. That is what is essential. So that is the reason biosafety will devise protocols how to work in the laboratory, how to transfer the organisms from the laboratory, how to dispose of once your organisms are used, how to dispose of them. That also it is going to design. So this is one application, major application of our biosafety. What are the other applications? These are the other applications for the same. It is going to help you design the control measures for hazardous organisms. How will you control it? Because you will learn eventually with biosafety units how to do risk assessment. So when you don't understand the organism's risk, firstly you are going to check how far they are risky, whether they are severely risky, moderately risky, they are mild, or they are just harmless. So as per that you are going to deal with these organisms. So then only you understand how to design the control mechanism against the organisms. So it will not only do the risk assessment, it will make some standards also for risk assessment which are universally accepted, some common procedures, protocols for risk assessment. It will also help in identification of source of risk or the contamination, and then it will design the control measures for it. So that is how no biosafety does lot of um, we should say a very basic fundamental work. Before you actually start your experiment, you have to begin with the containment level to be established in the lab. So it is a very basic protocol. From where you can actually do your genetically modified procedures. So it helps in setting standards in relationship to how are you going to handle your genetically modified organisms in the lab, off the lab, okay, during the transport, and also how you will be packaging them. If you have to carry it somewhere or you have to store it in your laboratory, how are you going to package it? So there will be a lot of protocols designed after making a judgment over the risk assessment given by it. So that's the basic idea behind this particular biosafety. So where we need this biosafety from our point of view, from our biological sciences point of view. So it's not that it's only biosafety is only applicable to micro organisms and only microbial science students or those who are into microbiology, they only need biosafety. It is for everybody who deals with any kind of organisms, wherever they are. So they can be a part of life science, they can be somewhere else. If they're dealing with some organisms which have a potential risk, they need to set up the biosafety. So the first need is the lab that has hazards of processing infectious agents. So lot of laboratories who are involved in experimental procedures or testing procedures wherein these infectious agents are present, they are dealing with hazardous waste, they are dealing with hazardous organisms, and they need biosafety protocols. Another thing is accidental threats to workers and environment. So you need to establish this. Otherwise, whenever there will be some accidents happening in the laboratory, how are you going to manage them? Suppose by chance, even if you have a very good no containment established inside your laboratory, somehow the organisms escape from the lab and you have an accidental exposure to some worker or the nearby environment, there has to be some emergency protocols which can help control the organism's threat on a large scale. So for that, biosafety is needed to have adherence with safety regulations while dealing with highly infectious agents. So we need that people should follow this. So you have to make stringent rules that people working in certain sectors, they must follow certain protocols. So for that also, for no bringing the regulation also, biosafety is needed. It will design protocols, it will design safety measures, it will design the standard operational procedures, that is SOPs, rules, regulations, and guidelines, laws, and acts. So you have a complete unit which will be taken care of, further in the paper, wherein you will be studying different types of laws and regulations and guidelines. For example, you will be studying Cartagena protocol, from which you have the guidelines made. RDNA technology has its own biosafety guidelines. So those will be the ones which you will go through. There are guidelines given by WHO as biosafety manual. So you will be studying those. So if you have gone through your syllabus properly, and if you recollect, there are hyperlinks given for the protocols. So those are the ones which eventually you will be studying in the units coming forward after this particular unit. So Chani madam will conduct the class for that, right? So this is the basic application and need of biosafety. Right now, we move on to another units. So we have almost done with our first unit's basics. That's already been done.